One of the oldest and most central concerns to the study of life is to organize and describe its diversity. Being able to agree on a name for an organism is a crucial requisite for any type of research to follow, especially when studying an organism's evolution. It can be easy enough to tell the difference between an eggplant and a sparrow after 1.5 billion years of evolution since the divergence of plants and animals, but when we narrow our focus down to groups of organisms that split more recently in evolutionary history, the biological differences that delimit one species from another are not always so obvious to observe and require special scrutiny. My research this summer dedicates such scrutiny to identifying species of lichens, which are organisms you have likely encountered growing on a rock or a tree. A lichen is a symbiotic organism composed of a fungus and a photosynthetic algae or cyanobacteria. The two live in partnership and benefit from each other in a lifestyle called mutualism. My project is concerned with recent collections from Australia, New Zealand, and Chile to study the evolutionary history of two closely related genera, Stereocolon and Lipraria. I've employed three techniques to the aim of identifying these collections to species. The first method involves traditional attention to morphology, which refers to the physical characteristics of the lichen, even looking so closely as the microscopic level to determine significant features that have been associated with particular species. This is useful when the organism of study has features discernible enough to be adequately characterized, like with the genus Stereocolon, which tends to grow a complex vegetative structure. But that's not always the case, as with Lipraria, which only appears as a bluish-green dust on rocks and trees, so indiscreet that not even a microscope can help with species identification. That's when we employ the second method, chemistry. Considering that lichens grow exceptionally slow and very exposed, one might think they're vulnerable to animals and the elements. But millions of years of evolution have outfitted them with chemical defenses that offer UV protection and ward off herbivores. Since different lichens produce different chemicals, called secondary metabolites, we can identify species of lichen by classifying which chemicals they produce. Using a method called thin layer chromatography, or TLC, we soak the specimen in acetone to extract many of the chemical compounds, then apply a drop of the solution to a thin absorbent layer of silica to separate the component chemicals into colored bands via capillary action. But even the chemistry of lichen compounds may not provide enough detail to delimit species, which leads us to consider differences in DNA. To do this, we isolate and sequence a small region of the genome called the internal transcribed spacer, or ITS. Since all specimens that we study have the same ITS region in their DNA, we identify small differences in the sequences to assess the degree of relatedness between lichens. The ITS also acts as a species barcode. By comparing our ITS sequences to massive genetic databases online, we can, for example, easily verify whether we have Stereocolum loricatum or Stereocolum vesuvianum. I'm doing this study to better understand the distribution and biology of lichens, such as Lipraria and Stereocolon, and once we have a better understanding of where certain species occur and how similar they are to their close relatives, we can continue to inform the broad study of life and the mechanisms of its mutable form.